All right, so last part of chapter 2. We started off talking about carbohydrates, then we switched to lipids for our organic molecules. Now we're going to talk about proteins, and finally we're going to talk about the nucleic acids. So proteins. Major components still include carbon, hydrogen, and oxygen, but a new major component is actually nitrogen. Some minor components can include sulfur, phosphorus, iron, and iodine. Something that's kind of unique with a protein, their molecular mass can be huge. So sodium chloride is a molecular mass about 58. Glucose, which is the C6H12O6 that we've been talking about, is about 180. Proteins can be thousands to several million atomic units in molecular mass. The thing to keep in mind with proteins is they can form complex machinery. They can actually form things with moving parts and pieces. So, the functions of proteins, the functions of proteins, the functions of proteins, the functions of proteins. Think it's important? Good. 1, regulation. We talked about enzymes toward the middle of the chapter when we talked about catalysts. Catalysts and enzymes are pretty much the same thing. They control chemical reactions and they have moving parts. We talked about me taking my hands and taking the nut and the bolt and putting them together, right? Hormones can also be proteins, and they can regulate quite a few of your body's physiological functions. Insulin is an example. Growth hormone is an example, and they are regulatory molecules. Transport. They can help transport things in the watery environment of the blood, and they can control movement of things into and out of the cell. They can actually create doorways in your cell's membrane that allow things to go back and forth between the outside and the inside, or the inside and the outside. As well as that, they can act as transporters, almost like a taxi cab, that something can get into that particular transport protein and then it takes it to its destination. Protection. I'm sure most people know what an antibody is, especially in this day and age with COVID going around. Antibodies are a protein. That is what they're made out of. And it does provide you with a certain amount of disease protection. The complement system is also a set of proteins that protects you against foreign invaders. Contraction. So actin and myosin are protein fibers that basically crawl on each other. And when one crawls on the other, it makes your muscles shorter. And when it lets go, your muscles relax and they lengthen. But that ability to crawl and use kind of leverage to get that muscle to shorten is actually a protein function. Structurally, they are also the collagen fibers that help to make our tissues more stable. Keratin that gives our hair and nails strength is another type of protein. And finally, they can be broken down to make energy. Now, I'll admit your body will go through everything else before it gets to protein as a way of collecting energy because it doesn't like doing it, but it can. So remember I said pound per pound, lipids give you twice the amount of energy as sugar. With protein, it's about equivalent. You get 4 kcal per gram of sugar, you get 4 kcal per gram for protein. So the building blocks of proteins are the amino acids. This is an amino acid. Now I want you to notice something. There's a central carbon, an amine group, a carboxyl group, and a hydrogen. Every amino acid that there is, all 20, have these same parts. The only thing that changes is this little dude right here, this R group. So, you know how in an algebra equation, the X is the unknown? In an amino acid, the R is the unknown. It changes from amino acid to amino acid. Peptide bonds. So between each amino acid, we have what is known as a peptide bond, where you take the carboxyl group, the carboxyl group of one amino acid and bond it to the amine group of the next amino acid. When you make that bond and take that water out, this bond between the carboxyl and the amine group is called a peptide bond. And it doesn't matter where you do it. It can be in a protein, but it can be in anything else. You put a carboxyl and an amine together, it's a peptide bond. When you have several amino acids bound together this way, though, you will call it a polypeptide or a polypeptide. You can call it a polypeptide depending on what you're building. I guess how big and how many of these amino acids you have in the chain. These are the 20 amino acids. Do not memorize them. That is not what I'm asking you to do. What I do want you to notice, though, is all of them have the same base. Okay? Every single one of these have the same base up here. You've got a carbon with a hydrogen, a carboxyl, and an amine. Remember I told you the R group is the only difference between them? Remember I said that? These down here, that's a terrible color, hang on. Try this. These down here are the R groups. And you can see they vary pretty dramatically. You have R groups that are as simple as, let's say, glycine. Glycine right here, where it's just a hydrogen all by itself. But then you've got these huge long chains, like what you see in arginine down here, or you've got things like rings, like you see in histidine over here. That R group varies quite a bit. Now, if I'm putting these together like a pearl necklace and the pearls are different shapes, it can actually kind of change and bend that pearl necklace, especially if I've got these huge ginormo pieces that are being put into this chain. So again, not asking you to memorize it, I'm just showing you. When I was in school, nobody ever told me that that's the way that was. And it never occurred to me that the base was always the same. And the only thing I had to really worry about was the R group. So I wanted to show you that. So the 4 levels of protein structure. You've got primary structure, secondary structure, tertiary structure, and quaternary structure. Primary structure is very simple. What's the sequence of amino acids? That's it. Arginine, glycine, aspartic acid, glutamic acid. It's literally, tell me the chain of amino acids. What's its sequence? So it consists of a chain of different amino acids. Now, the secondary structure is also called the local structure, okay? And it basically involves small regions of that long chain of amino acids that are held together by hydrogen bonds. Remember our hydrogen bond where the hydrogen had a partial positive, so it's attracted to a partial negative? You have 2 structures in the secondary structure. You've got this little dude right here called a beta pleated sheet. Kind of looks like a paper fan, the kind of fans you made when you were in school. And then you've got this thing right here called an alpha helix. Those are the 2 secondary structures that you see. Now keep this in mind. It's a local structure. It's not the whole thing. So if you look down here, see this strip of blue and this strip of blue here? That's what they look like. If you were to look at them up close, you would have a fan here and you would have a fan here. So it's a local structure. It's just a little piece of it. The alpha helix is a local structure. It's just happening right there. Now, admitted, there are other alpha helices here and here and here, but it's not the entire thing. It's just a piece of it. Secondary structure. It's a local structure held together by hydrogen bonds. Now, the tertiary structure. Tertiary structure you can think of as the three-dimensional structure. In other words, how does that entire pearl necklace fold together? What shape does it have when it's folded the right way? Okay. So, what does it look like in 3 dimensions? The quaternary structure. That's when you take multiple tertiary structures and put them together. So if you look at this is actually just this orange one right here. Okay? You can tell that I've got another one over here, another one over here, and another piece over here. The thing with proteins is that they make these big complex structures that can have moving parts. If you think of a car engine isn't just one big chunk of metal. It's got different parts. It's got pistons. It's got fan belts. It's got, not shock absorbers. What's the word I'm looking for? Spark plugs. It's got all of these different parts and pieces to make it run. Quaternary structure means you're taking multiple tertiary structures and you're making a bigger, more complex structure. This structure specifically down here is actually hemoglobin, which is the thing in your blood that carries oxygen. So, the enzymes. Remember with catalyst, we said that it lowers the activation energy by putting stuff together the way we need it to come together. So a protein catalyst increases the rate of a chemical reaction without being changed itself. I could take the nut and the bolts with my fingers, put them together, and my fingers didn't fall off. They didn't fuse to the nut and bolt. I could do that and go and do it again, right? Now, with an enzyme, which is just a biological catalyst, the three-dimensional shape is essential for it to function. You have to have the right shape to your enzyme if you want it to put things together the way you want them to. There's something called the induced fit model, okay? So if you look over here, here's my enzyme. This is what I want to come out of it, but here it's not even kind of close to what I want, right? So when this comes in and this comes in, I change the shape of this protein to get me what I want. I induce the fit fairly significantly to give me my product. I have to be in one shape. When my reactants come in, I have to have kind of a change to my final shape to get my product. Just like catalysts, which, by the way, catalysts and enzymes are pretty much the same thing, they lower the activation energy because they orient the reactants in such a way that the chemical reaction is more likely to occur. The reason I could use my fingers to put that nut and bolt together is because I can put them in the right position to get them to spool, right? Same thing. So, an enzyme binds to reactants. It combines the reactants, okay, and then it releases the product so that it can do it all over again. Enzymes, just like catalysts, are capable of doing this multiple times. It's not like it's a one-trick pony. It does it once and then it dies. There are some things like cofactors or coenzymes that basically are required to get an enzyme to function. Think about it this way. If I want my car to work, what do I need? I need a key, right? Enzymes are expensive. They're expensive to make. I don't want to make an enzyme and have it work for, one time and then I tear it apart. That would be like me saying, I'm going to go and buy a car. Okay, I've used it once, and throwing away the key. What are you doing, right? That's expensive. So what do you do with your car? Do you leave it running 24/7? No, you have a way to turn it on and you have a way to turn it off. Cofactors and coenzymes are kind of like that. They are little pieces that I don't want my enzyme running right now. You take it out, it shuts it down. When you're ready for it to work again, you put it back and it starts running again. Why run an enzyme if you don't need it? And by the same token, I don't want to have to make new enzyme every time. I don't want to have to go out and buy a new car anytime I'm leaving my house. So what do? I turn it off when I don't need it. I turn it on when I do. ACE. If you see this suffix at the end of things, it is usually going to be an enzyme. So for example, protease. It's a protein enzyme. Lipase. It's a lipid enzyme. So the nucleic acids, DNA and RNA, they are also made up of carbon, hydrogen, and oxygen. Nitrogen is also a major player, but so is phosphorus, okay? DNA is one of our nucleic acids, and it carries our genes. It carries our instruction manual. RNA actually has quite a few jobs, but it does encode amino acid sequences for proteins. The building blocks of our nucleic acid, remember the building blocks for protein was amino acids? Well, the building blocks for our nucleic acids are called nucleotides, okay? So we've got DNA. Hang on. We've got DNA. DNA, also known as deoxyribonucleic acid, comes in the form of a double helix. It looks like a twisted ladder, okay? RNA, or ribonucleic acid, actually looks like just half of a ladder. It does not come in a double-stranded variety in your body. The basic structure of a nucleotide is a 5-carbon sugar in the middle, a nitrogenous base on one end, and a phosphate group on the other. Okay, so if I were to draw a stick figure of a nucleotide, that's what it would look like. Sugar in the middle, nitrogenous base on one side, phosphate group on the other. So what's the big difference between deoxyribonucleic acid and deoxyribose and ribose? That. Deoxy, without oxygen. There's one less oxygen in deoxyribose than there is in ribose. Now I know you're probably going, well, that's not a lot. If you think about it chemically, it kind of is. The nitrogenous base. So this that I just showed you was this sugar right here. Now we're going to talk about this guy over here, the nitrogenous base. We have two different types of nitrogenous bases. We have pyrimidines and we have purines. And the thing that makes them different is their structure. Purines are a double ring structure. Pyrimidines are a single ring structure. Okay? Purines include guanine, which is found in both RNA and DNA, okay? And adenine, which is found in both RNA and DNA, okay? It's found in both. Cysteine is one of our pyrimidines. It is found in DNA and RNA also. Now, the last two are kind of specific. Thymine is only found in DNA. Uracil is only found in RNA. So if you see a sequence and you see uracil in that sequence, you know you're looking at RNA because that's the only place you find it. If you look at a sequence and you see thymine, you know that you're looking at DNA because thymine only happens in DNA. So my genetics professor, when I was working on my undergrad, always said that ags were pure. So A and G are purines. Everything else is a pyrimidine. So like I said, with DNA, you've got adenine, guanine, cytosine, and thymine. With RNA, you have adenine, guanine, cytosine, and uracil. Again, this one is unique. This one is unique. You don't find it in the other one. Yeah. RNA is single-stranded, half a ladder. You've got the side of the ladder and you've got half of the rungs here. Okay? DNA is double-stranded. Here's one half of the ladder, here's the other half of the ladder, here are the rungs attaching to each other. Something that I do want you to notice here. Look at the sugar here versus the sugar here. They are actually going in opposite directions. We say that they run anti-parallel. Okay? They run in opposite directions of each other. So DNA, like I said, is a double helix. It's the whole ladder, but it's twisted. Vertically, this way, the nucleotides are held together by a covalent bond between the phosphate of one group to the next. Okay? So you've got a covalent bond. I'm not doing hydrogen bonds here. Horizontally, though, this way, these are actually hydrogen bonds holding your DNA together. Now, I do want you to notice that we have something called complementary base pairing. Complementary base pairing, meaning that I have a specific partner. Okay? Adenine always goes across from thymine. You can see that there and you can see that there. Adenine and thymine are always going to be partners. Guanine and cytosine are always going to be partners. You can also see that guanine and cytosine have 3 hydrogen bonds between them. Adenine and thymine only have 2. If you put them together the wrong way, your DNA either ends up with a huge bump in it or a huge dip in it. Instead of having equally sided ladder sides, you end up with something that looks very odd, okay? With RNA, there's also a complementary base pair. Cytosine and guanine still go together. That never changes. But since RNA doesn't have thymine, adenine has to be paired with uracil. The 2 opposing strands, as I said, run anti-parallel to each other. Look, the sugar's pointing this way on this one and this way on that one, meaning that the sugar phosphate backbone of one strand runs in the opposite direction of the other. One is in the 5 to 3' direction, one is in the 3 to 5' direction. It has to do with the carbon number on that sugar. That's the 5' to 3'. 5' to 3' is the one that is technically running in the right direction. It would be like reading a book from right to left instead of left to right. Within the DNA sequence of bases is a code that stores information used to determine the structure and function of cells. So a gene is basically a sequence of DNA that directs the synthesis of an RNA that will become a protein. This is literally your recipe book for how to make things in your body. Adenosine triphosphate, ATP. This is our fuel in our body. You've got a ribose sugar, an adenine, and instead of just one phosphate, now you have 3. Okay, adenosine triphosphate. Now, the cool thing is that this 3rd phosphate here gives you a lot of energy. So there's potential energy stored in that 3rd phosphate group that is essential to living organisms. It provides the energy that we use to do almost every chemical reaction in our body, including moving our muscles around. Okay? So, something you have to understand with ATP. ATP is made by taking an adenine diphosphate plus a phosphate plus energy to make ATP. We can break down glucose, fat, or protein to release energy, and then through a series of oxidation reduction reactions, we end up with ATP, that fuel source. ATP is used to provide energy for all of the chemical reactions in our body, and, like I said, to drive cell processes like muscle contraction. ATP is often called the energy currency of the cell because it is both capable of giving and taking energy. Yes, I can break it down to get energy, but it still costs me energy to make it. So I always talk about I always ask, do you pay to have your job? And most people don't raise their hand. And I'll say, okay, do you pay for gas so that you can get to your job? Yes. Do you make car payments so you have a car to get to your job? Yes. Do you pay for clothes so that you have clothes to go to your job? Yes. So technically, you pay to have your job. I know I pay to have my job. I'm not going to show up to school wearing my PJs, right? So, with that in mind, my job does cost me money. Now, what's the goal though? The goal is that it costs me less money than the money that I make, right? I want to make more money than I'm spending on my clothes and my gas and my car payment and all of that. Yes? Okay. So that's kind of what we hope for too. When we make ATP, yes, we are expending energy, but hopefully it's less energy than what we're getting out of the ATP that we're making.